Okay, in the last videos we learned about monohybrid and dihybrid crosses. So we're going to apply those concepts to incomplete and codominance. It is very important that you watch the previous two videos before you even attempt watching this video, because you need to know how to do Punnett squares first. So, incomplete dominance. So, incomplete dominance occurs when neither of the alleles can completely conceal the other allele. So what that is implying is you don't have one trait overtaking another. So you don't have complete dominance, which is why we call it incomplete dominance. The previous two, when I talked about mono and dihybrid crosses, we focused on complete dominance, where one allele will completely overtake the other. In this scenario, you kind of have blending of traits. So the example we have here are flowers. So in this flower, you have a red and a white flower. Now, if you mix a red and a white flower, you can get pink flowers. Okay, so that is showing you incomplete dominance because the red does not overtake the white. So the key with this one is they can do these Punnett squares multiple ways, and you may see any one of these on a test. So sometimes instead of using letters, they actually use numbers to represent the trait. So for example, you might use a 1 to represent the darker trait, I don't know, whatever trait you want it to be. So for example, red, you might have C1, C1. So C is representing color, the idea that it's color. 1 is representing the fact that it's red. White would represent color 2, so C2, C2. And then if you had, sorry, I need to get my pen back. So if you had C1, C2, that would mean you have a red allele and a white allele, meaning that it would be pink. Okay, so you can use numbers to represent these traits, or you can use letters like you see over here. So as you can see, this one right here, or could be, so let's just look at this one actually. So you can see this red one, this red flower, CR, CR, so color red, color red. So this is purebred red. Then you have CRCW. So CRCW means you have one of each of the color alleles representing blending of traits. So this can only be used when you have incomplete dominance. So the only time you would have white is if you had CWCW. So these are the two ways that they represent incomplete dominance. So some examples, and you can click these links on the digital version of the notes package, which is back on your monohybrid post. So familial I can never say this, hypocholesterolemia, Tay-Sachs disease, hand size, and hair type are all examples of traits that are caused by incomplete dominance. And as you looked up here, also flower color can be another one. Sickle cell anemia, this is an important trait. You see this quite a bit on tests. So this is a disorder in which the hemoglobin, which is in your red blood cell, okay, this is what allows the oxygen to bind to it. So if you didn't have the hemoglobin, oxygen couldn't bind to the red blood cell. So in sickle cell anemia, that hemoglobin creates needle-like crystals that are going to distort the shape of the red blood cell, making it very hard to move through the blood vessel. So normally in a red blood cell, you have this biconcave shape. So it kind of looks like this from the side, almost like a weight. Okay, so this makes it easy to flow through blood vessels. In the sickle cell anemia patients, your red blood cell is distorted. Okay, so it might look something like this. Okay, this is harder to move through blood vessels than this sucker. Okay, so this is very bad. This will cause extreme pain because you have something moving through a blood vessel that's very hard to get through that blood vessel, so you'll feel pain. So here is an example. So we have two people that have the sickle cell trait. Okay, doesn't mean they have sickle cell, they just have the sickle cell trait. So if you look at the example down here, it tells you what their genotype is or their alleles. So you have HB, A, and HBS. S is representing the fact that you do have the sickle cell trait, but it is a recessive trait. Okay, so sickle cell is recessive. So this person doesn't actually have the disease. Okay, this person. So you have two people that are heterozygous for the trait. Okay, they don't have it. So this example is known as a heterozygous advantage, specifically for sickle cell anemia. The reason why they call this an advantage is because patients that are heterozygous for sickle cell are actually resistant to malaria. Okay, so that's a good advantage to have. But the problem with it is if you are heterozygous and you marry someone and have children with someone who is also heterozygous, you can pass on the trait to your offspring. So as you can see in this Punnett square up here, you have a 25% chance of passing on sickle cell anemia to your child. So this child would have sickle cell. So they're not resistant to malaria. You would only be resistant if you had one of each allele. So this kid and this kid would be resistant. This one wouldn't. Okay, let's do some examples with these Punnett squares. So in Japanese 4 o'clock plants, so red is incompletely dominant over white, represented by little r. The heterozygous condition, big R, little r, results in plants with pink flowers. Okay, so you know that it's incomplete dominance because you get pink flowers when you have heterozygous. For each of the following, construct a Punnett square and give the phenotypic and genotypic ratios of the offspring. So we have a red plant and we have a white plant. Okay, so the red plant, since this is incomplete dominance, the only way that you're going to get red is if you have two of the dominant alleles. Okay, so you can represent this by CR for this red plant, or you could say that this is trait 1 and this could be trait 2. So, or you can do C1, your choice how you want to do it. Some people, since all of your tests are multiple choice, some people can just do this and know that that's red. Okay, it's more preferred if you do these two, but I'm okay with whichever. Okay, so I'm going to stick with this one because I can see the R's and what they represent. Now we have a white plant, so CW, CW, I need 2 for it to be white. So I need to cross these two. So CR, CR, CW, CW. So it doesn't matter which one comes first because we have an incomplete dominance. So we have CR, CW, CR, CW, CR, CW, CR, CW. In some of these Punnett squares, you can probably look at your parents and say, oh, well, they're all going to be this. And that's fine, especially if you're doing a multiple choice question. Okay, so my genotypic ratio, 100% of the offspring. If you mate a purebred red and a purebred white, you'll get 100% CR-CW. Okay, so again, a genotype is a representation of the alleles. Now, my phenotypic ratio is what you see. So what does this show you? So R and W, or R heterozygous, is representing pink. So you have pink flowers. Okay, I want you to try B and C on your own. Okay, and then check with the answers to make sure you got them right. I'll do one of these and then move on to codominance. So in some cats, the gene for tail length shows incomplete dominance. Cats with long tails and cats with no tails are homozygous for their respective alleles. So that's telling you that long tail has to be two of the same allele. And then, this is saying, two of the same allele. Cats with no tails are homozygous for their respective alleles. So these will be two of the same as well. But they'll be different traits. Cats with one long tail allele and one no tail allele have short tails, so kind of halfway between. For the following, construct Punnett squares and give phenotypic and phenotypic ratios. Okay, so this doesn't tell me what the letters are, okay? So let's do the numbers for this one. So long tail, so now I'm going to use big T to represent tail, okay? And I'm going to put TL to represent long tails. Again, you can use whatever. Actually, we're going to use numbers, like I said, and I started using not numbers, so my bad. So I'm going to change it to T1 to represent long tails. Cat with no tails are represented by T2, so that's the second trait. And then one long tail and one short tail allele, one long tail and one no tail would be one of each, so T1, T2. Okay, so we have a long tail cat, so that's this one. So we have T1 is mated with a cat with no tail. Okay, so no tail is T2. So we get T1, T2, T1, makes me want tea. T1, T2. So all of our offspring will be T1, T2. That means 100% of my offspring will have short tails. Okay, so this is very similar to the first one, so I'm going to do another example. So I'll do B. So a long tail cat, so that was T1. So T1 with a short tail cat. So that was T1, T2. So here you get T1. So that's a long tail. Here you also get T1. Down here, this T2 is carried into both, and you can only pass on T1. So T1, T2. So we get 50-50. Okay? So you're going to want to do 2 by 2, but remember, you have to reduce it. So right away, I know it's a 1-to-1 ratio. So 1 T1 to 1 T1 T2. So what did the T1 mean? So you get 1 long tail to 1 short tail. So it's a 50% chance of getting offspring if you made a long tail and a short tail. Try C and check the answers. And now we're going to talk about codominance. Okay, so codominance occurs when both alleles are fully expressed. So you have nothing overtaking another. So if you can look at this picture, horses or hair in a lot of animals will exhibit codominance. So here you have brown and white both being dominant, which is why you see some hair that's brown and some hair that's white. So one color is not going to fully overtake the other. So you're going to do this the same way you did the previous one. But now I'm going to switch it to different letters to represent the different colors. Okay, and you can do either or when you're doing your assignment, because this is all going to be multiple choice since we're online learning. So in certain fish, blue scales and red scales are codominant. When the fish has a genotype BR, it has patchwork of blue and red scales. So what happens if you breed this fish with one that only has blue scales? So it's saying that you are breeding this patchwork one. So that's saying you're taking this one. Okay? And then only blue scales. Sorry, I didn't mean to go into the word, but only blue. So I would assume that this B is going to represent blue. So I'm going to have a fully blue scaled fish. So in my Punnett square, I get BB, BR, BB. Oh, look, it's my initials. And then BR again. So looking at the offspring here, this is representing blue, blue, and this is patchwork. Okay, so you have some that are blue, some that are red scales. Some that are blue, some that are red. So it's a 50-50. So it's a 1-to-1 ratio again for my genotype. Okay, one represented blue and BR represented patchwork. Okay, try number 2 on your own and check if you did it right. Last thing I need to talk about in this video is Walter Sutton. So Walter Sutton studied synapses, so that's in your neurons, and the migration of sister chromatids during meiosis 1 and meiosis 2. Okay, so that's talking about the movement of the chromosomes during cell division, and specifically meiosis, so the creation of the gametes, for example. He came up with the chromosome theory of inheritance. So this stated that genes are carried on the chromosomes, which segregate independently during meiosis. So basically, they were pulled apart during meiosis, and which we know is actually the case. If we think back to the last chapter, they are actually pulled apart. Sister chromatids, specifically in meiosis 2, the sister chromatids are pulled apart. So he is partially true on that, and we'll talk about in the next video how his theory was built upon. Okay, do those practice, check if you're right, and then watch the next video.